So now we come to the final talk of this session, which will be by Meghana Krishnadas from NCBS, and it'll be another talk on plants.
Good morning, everyone, and thank you so much. It's a pleasure to have this opportunity to present my work here at the Academy. And what I'm going to present is going to have a slightly different flavor. I was from the talks that you have seen so far, in that you're not going to see a lot of genetic data or a lot of molecular data. This is something that we are sort of considering getting into. But I am by training and by a lot of my work historically has been that of community ecology. So which means that instead of, I sort of look at patterns and processes at the scale of the here and now. I look at what is governing how communities are assembled and how different species come together to form a community and stay there. So the way I look at it is many of the evolutionary outcomes that we see at, if you're looking at deep time or larger spatial scales, things that we broadly characterize as niche differences or, speciation that has taken place. The way I see my work is I look at what is the ecological stage on which many of these evolutionary processes seem to play out, and can we look at what's happening here and now in terms of what is shaping these many species coming together to form a community. And so that's the kind of flavor in which you'll see much of what I'm presenting today, but also historically what I have done. Now, I have worked mostly in the Western Ghats. That's where I have a long-term project going on, and that's where a lot of my work has looked into the diverse communities of trees that we find there, and how do we explain so much tree diversity and what maintains this diversity, both in terms of the response of plants to their abiotic environment, but also in terms of the biotic interactions that they have with say insects and pathogens. And this is not work that I will present today, but this has been the broad focus of what's been going on in the lab. Now, because of the situation or the geographic situation in which we are, this event is being held, which is IISER Tirupati, I thought it would be nice to present some of the newer work in the lab that we have started in the Eastern Ghats. And this is looking at how plant communities are assembled and what seems to be happening to plant communities in the surroundings here that we'll see today. And this is something that this is what I will focus on in terms of my talk today. Now, ecosystems across the globe are facing a myriad of changes, and a lot of this is because of human activities. So humans, in their quest for space, resources, have altered ecological systems at large spatial scales, which compare with that of what meteorites and volcanoes have done in the past, in some sense. And this has led to issues such as fragmentation and degradation of habitats, large-scale nutrient deposition, species invading into habitats where they've not been seen before, and of course the looming specter of the climate undergoing some massive changes. And these global environmental changes, individually and interactively, are reshaping ecological communities at massive scales. Now, to understand how these communities are going to respond to these changes that are happening fairly rapidly, today more than ever, we need to first understand how do these ecological communities form, come together, and stay in the first place. So what is it that governs where, how these many species assemble, and therefore can we then predict how they might disassemble or reassemble in terms of what happens with ongoing changes to the situation where they find them. Now, biotic assemblages vary in we know that they vary in space and time, and this comes about because of certain processes. You may have evolutionary processes creating a meta community or a historical pool from which species sort of have to disperse into these various smaller spaces where we eventually find them and study them as ecological communities. And even after they disperse, in order to establish at a place, they have to go through an environmental filter, which is that they need to be suitable to these particular environmental conditions. And you saw some reflection of that in terms of what Robin was saying, what is a climate specialization or habitat affiliation. So not all species can be found everywhere. And once they do come to a point where the climate is suitable for them, they still have to contend with other species in that milieu. They need to have they engage in these biotic interactions, which can be competition with species in the same trophic level, or interactions such as pollination, herbivory, and those which are occurring with other species, dispersal, which allow them to then stay in the community and form these diverse communities, as it were. Right? So the broader theme of my research or the research in the lab is to understand some of these processes. Like when we look at ecological communities in nature, when we look at multiple species, can we understand which of these processes seems to be largely shaping these communities, and then do smaller scale experiments to tease apart some of these processes. But the idea is, can we then help detect or use this understanding to help detect how biodiversity may change or across space and time with ongoing pressures that they are facing. Now, the reason species are not all species are found everywhere, and species seem to prefer one place over the other, is because they differ in their inherent features. And this is known as traits. These are morphological, anatomical, or physiological characteristics in which govern functional responses of species. It might be how they respond to temperature or water availability, nutrients in terms of plants, right? So how they use resources, how well they tolerate stresses, or how well they can compete with each other or have certain interactions that they engage in. But the thing is, the role of a trait of any particular functional feature can vary with the context in which you look at it and the scale at which you look at it. The same for plants, for example, there is a trait which is called specific leaf area, which is the amount of leaf area per unit gram of leaf tissue. And it's considered to be a really important biogeographic trait in terms of how specific leaf area is distributed at different parts of the world. However, the role of specific leaf area may be very different when you have plants that are in the Western Ghats, which is a fairly wet, humid ecosystem, versus the Eastern Ghats, where it has to deal with a different kind of stress. The same value of the leaf area might be there, but they might be playing different roles in the way they are filtering species in these two different, very different ecosystems, right? And in this context of trait-mediated assembly of communities and trait-mediated understanding of ecosystems, surprisingly, dry ecosystems across the world, and particularly in the tropics, have been undersampled and underrepresented. So a lot of our understanding of how traits or functional features are shaping communities is based from only a subset of the kinds of communities or the kinds of biomes and climatic conditions that we see around the world. And this is a gap because many of these tropical dry ecosystems are actually under fairly severe threat. Many of these species are considered, they're already very harsh climates, they have really strong dry seasons, and many of these species are considered to be at the edge of their ability to tolerate some of these conditions. So today more than ever, we need to know how traits are determining what's happening in these understudied systems, right? And we know that for these tropical dry systems, the species have to be uniquely adapted to withstand these conditions that I mentioned, right? And even within this fairly harsh system, we know, when we start zooming in, we know that there is environmental heterogeneity, and we know that species differ in where they are found. So there is some knowledge that there is environmental filtering happening even at this scale, but what is it that's driving these filters, and how do the traits mediate some of these changes to the communities is still not very well known for some of these systems. So this is the broad context in which we started our work in the Eastern Ghats. This is work that I started while I was in CCMB Hyderabad, and we thought that we would take this fairly large spatial scale of 6,000 square kilometers encompassing 3 protected areas in the Eastern Ghats. This is a forest to savanna transition that happens from the south here to the north, which the southern parts are Gudla Brahmeshwaram Wildlife Sanctuary, which is relatively more moist, and as you go north, it becomes much drier and more open savanna. But there was a fairly short gradient of temperature and precipitation, but a lot of variation in terms of topography, and which led to variation in terms of like hills, valleys, and slope, which led to microclimatic or microenvironmental variation that we considered would be important. And the first thing we wanted to see, does woody cover and tree species composition change across the system? And we did that laying out these many vegetation plots across this very large landscape to characterize what the tree community looks like here. Right? And when these sites, as expected, differed in their climatic conditions, we looked at the macroclimate, which is here. We pulled this data from remote sensing, from publicly available databases that are there, and we found that the sites that we studied could be discriminated by sites that had that were warmer, slightly warmer, but surprisingly wetter, but had stronger temperature seasonality. Which was the this was the set of variables that were most strongly discriminating the sites, right? But we also had microenvironment, which seemed to play a pretty strong role. But not as much as the macroclimate in terms of discriminating sites with moist soils and high phosphorus, and some sites which had low nitrogen but were acidic in nature. Now, what I'd just like you to note here is that overall, even though the macroclimate gradients were really short, actually it explained a lot more variation among the sites than microclimate gradients wherein there seems to be a lot more heterogeneity. And this is something, the significance of this is something we realized when we started looking at what was happening to the communities that assembled along these gradients. And in short, it seems like macroclimate did not seem to matter so much for the diversity of tree communities that you would find. And maybe that's not surprising. Like I said, the macroclimate gradient, although sites differ, it's still a pretty short gradient. What seemed to matter the most is that there was microclimate and particularly soil moisture that seemed to play a role in structuring the diversity of communities across the system. But surprisingly, usually you would expect that wetter sites, and this is what we see at a macroecological scale, wetter sites have higher diversity. We found the exact opposite pattern here, which is that wherever there was higher soil moisture in the dry season, we surprisingly had lower diversity. And most of these really low moisture sites were where we had much higher diversity, which sort of led us to believe that maybe it's because in this overall harsh macro environmental condition, most species are adapted to dry conditions, and that's where you find most of the large communities. There are very, very few species that are actually adapted to being able to survive in wet sites. And these are probably remnants from, historical remnants because of evolutionary refugia. And some of this is indicated in the functional diversity, which tells us how these communities are assembled. So if you factor out what the species richness is and just look at how much diversity there is in traits, we find the exact opposite pattern, surprisingly. And we find that these drier communities are comprised of species that are very similar to one another in their ecological features, in their traits. Whereas in the wetter sites, you find that there is more scope or more leeway for traits to play out, right? Which also seems to suggest that, some of these species might just be these remnant wet associated species that are there in these wet refugia, likely the valleys that are there in this otherwise very dry landscape. Okay. But we did see that, precipitation and temperature led to how species, the change in species as you go from one site to the other. So let me just walk you through this. The change in species or the similarity of ecological communities or the dissimilarity of ecological communities here can be characterized as something called an ecological distance. That is, we ran a model where we said that, okay, if there is this much difference in the community of trees that are found from one site to the other, could you put that together as a statistically significant ecological distance between these communities? And then you check to what extent this ecological distance relates to the change in, say, a variable like precipitation or maximum temperature of the warmest month. And how you read this is that as you go from a precipitation of 90 to 95, you don't have a lot of difference in what the community looks like, but when you go from 95 to 100, the community starts looking very different, right? Which suggests to us that at some changes in the temperature level, you're going to see big shifts in what species are going to be found, and that comprise many of these communities at these large geographical scales. But I'd like to draw your attention to one thing. If you look at the numbers here, the predicted ecological distance, which we found, this is actually in the data, right? And you look at what is the partial ecological distance that is factoring out all other variables, what is the ecological distance attributable to this variable alone. Macroclimate again seemed to explain, even though this curve looks very steep, the actual ecological distance or the dissimilarity in communities with respect to macroclimate is pretty small. And again, when we compare that to the microenvironment, primarily soil moisture again, but also soil phosphorus to some extent, which if you recall were the two variables that loaded the most dominantly onto that first principal component axis that was characterizing the microenvironment differences, we find that a much larger amount of the ecological distance, even after factoring out all other variables that are possibly driving it, is being explained by soil moisture, which gives you the idea that dry season soil moisture seems to be a very critical variable in this system at a scale which is telling you how communities are going to change as you go from one place to the other across this system. And some of these changes were driven by the response of individual species. Whatever I presented so far was this composite picture of what's happening to the community as a whole, right? But we wanted to get down to the base levels of, can this be explained by what different species are doing? Because eventually when we measure traits, we are going to measure that the phenotype at the level of species, and what we assume that species are getting discriminated based on their functional responses that are that traits somehow represent, right? And yes, we found that if you take the macro environment, their species clearly differ in their response, which is that there are some species, these values here, these species like warmer, wetter environments, but are also able to withstand stronger seasonality of temperatures. A bunch of species that don't seem to care much, and this is interesting because you have many species here which seem to be generalists, which is sort of what we expected, and which is also what would sort of tell us why is it that you have a very low functional diversity in many of these sites that we are looking at. But we do have some species here which seem to prefer, go in the opposite direction and prefer conditions that are cooler but can be drier. And this was something surprising and something that we would like to go into further, like what are these features that are discriminating species along these unexpected dimensions of temperatures and precipitation combinations, right? But again, the microenvironment seemed to be a much stronger discriminator of species responses across this habitat. What we found was there was a whole bunch of species that seemed to prefer moist soils and high phosphorus conditions, and many of these species, these genera that we know, are associated with riparian valley vegetation and things that have congenerics in the Western Ghats, which is wetter climate. And many of the species that we found here that were preferring the more dry sites were actually those that to be very dry associated species found in the Eastern Ghats and habitats around here, right? So again, we find that species are certainly responding to moisture and nutrient conditions, and primarily moisture. A lot of this was, we know that moisture was the main driver of many of these responses. But what we found was the traits that we chose to study were very poor predictors of these species' responses to the gradients. So even though we know that there was clear discrimination or differentiation among species in how they were reacting or responding to these gradients across the system, traits did a very poor job of predicting these responses, or at least the traits we studied. And that's why we found that even though there was compositional turnover in terms of the species, the dissimilarity models that I showed, again, the compositional turnover was not predicted by the traits that we chose in this system. And one reason could be that the gradients are too short, but we do see that there are clear differences among species in what they do. So the question is, what is going on here? It is, what is it about the species that might be able to explain this? The first thing you would think of is that, as I said, these traits are not really the traits relevant to the key drivers. So for example, if temperature is the key driver of where species are found, would thermotolerance or drought tolerance be a trait that we should be looking at? And these higher level, more synthetic traits, such as those of, the traits we studied are basically specific leaf area, dry matter content, bark thickness, and wood density. And these are traits that are more integrative. They are more at a whole organism level, and maybe they are not the ones that are really driving these mechanistic responses of species. However, when we did these experiments, we took some of these species, which are 15 of these species from this large study that we did across 48 odd species, and we said, let's choose species that are found only in dry sites or drier sites, slightly wetter sites, and the ones that are more broadly distributed. This is from the graph that I showed you for, how species are doing different things, right? And then we said, okay, let's measure these simple traits, but also a few more complex traits like what we call Turger loss point, which is a very good indicator of species response or their drought tolerance, right? And here with Turger loss point, what we would find is that, when the values here would suggest that these are more, less drought tolerant. And surprisingly, what we find is that by and large, when you are more drought tolerant among all of these species, you actually tend to accumulate better biomass, which means that yes, all of these species in this dry system would probably, the species more accustomed to drier conditions are going to perform better in general. But when you impose a drought treatment, which is this red line here, the difference among species in their roles or in their ability to accumulate biomass doesn't really change. So which means that Turger loss point as a whole, like macroecologically, is driving how species have come to be in this environment, but it's not discriminating species' response to water, a further loss of moisture from what we see. Surprisingly, leaf dry matter content, which is a simple trait, is still shaping or explaining how much biomass you're going to accumulate in dry versus wet conditions, where low leaf dry matter species, which are thought to be less stress tolerant and they are photosynthesizing more rapidly, they are actually, they as expected do well when there is plenty of water available, but their performance dips when there is drought, right? So clearly traits do matter at the scale of these experiments we do, but they don't seem to then translate into explaining what's happening at the macroecological scale of how these communities are assembled, right? And one reason that might be is because there is immense variation within species. And I seem to have taken a lot of time, but this is something that we've been excited about because this is fairly new from these systems, that if you have a system that is fairly homogeneous environmentally, do traits need to vary? And the answer is yes. We see that some of these traits, bark thickness, LDMC, leaf matter per unit area, and wood density, some of them have more variation within a species than they have among species, right? So in some cases like wood density, highly conserved trait, we don't see it. This was expected. But some of these other traits vary a lot, which might explain why we don't see this kind of, we don't know whether this is because of genotypic differences or because of plasticity in these traits, but this is why some species or many of these species can be found everywhere, right? But then we wanted to understand, is there still some role played by these, by the fact that there is so much variation and the fact that some species tend to vary more than others in their traits, right? Or the variation within species is really high. And what we find is that yes, intraspecific trait variation does relate to ecological properties, and a property such as abundance or the geographical dominance of a species correlates with intraspecific variation. If you have more variation in your phenotype, you tend to be more geographically dominant. This is kind of a consistent response in multiple traits. I won't go into some of the details of these parabolic responses that we see. We also see that when you have more variation in your traits, you tend to occupy wider niches. And this might be one reason that you're occupying many more niches, and that's why you are able to become more abundant. And we do see that if you are occupying more niches, you're more abundant, right? So these are various pieces of the puzzle. But the thing is, does it all come together? Because there are multiple ways that variation in a species trait can link to its eventual geographical properties that are macroecological, right? One is niche width, like I mentioned, that if you have an environmental gradient and you occupy wider niches, you may just be able to persist in more sites. So that increases your plot occupancy and that increases your abundance. But another thing that matters is how is the environment distributed in this place in the first instance, right? So if you have some species that are very close to the most common environmental conditions, which we term as low niche position. The blue here is showing the distribution of the available environments, or very broadly speaking, available niches, and you're very close to the most available niches. Low niche position, maybe that's how you become more abundant and has nothing to do with niche width, as opposed to something that is occupying very peripheral environmental conditions, right? So we put all of this together in structural equation models in which you can see which one of these factors relates to which one. Is it a direct relation or is it a mediatory relation? And we wanted to check how intraspecific variation goes on to shaping abundance, which is an important property of species, right? Is it through niche width or the niche position? Does that drive how many sites across an available landscape can you occupy? And does that then explain your abundance? And what we found is that two major traits seem to play a key role: bark thickness and wood density. Unsurprising in this landscape where fire is a key driver and bark thickness is a major variable that relates to how you respond to fire. But let me just summarize the key relationship, which is that bark thickness, the trait variation, although we know that it relates to niche width, that does not lead to any or has no mediatory role in governing the eventual macroecological properties such as how many sites you occupy or how abundant you are. What matters is how close are you to the most available conditions that are in a system. And this relationship shows that if you have wide variation, surprisingly, those species are actually occupying conditions that are very close to the environmental mean, and those are the ones that are occupying many more plots and therefore becoming more abundant. So how we are interpreting this is that if you have wide variation, you're going to have, you occupy many wider niches, and that allows you to occupy, to sort of sample much of the landscape. But really what matters is that you're occupying many of the plots by being close or being able to take charge or dominate the most common environmental conditions. And that is somewhere where the next step comes in, like being in these environmental conditions, these common conditions, do you have to vary a lot in trait to actually change how you compete with other species when you're in these conditions, right? Which is something we can't test in these sort of studies, but more detailed analyses of long-term forest dynamics or vegetation dynamics can let us know. But eventually what we see is that you occupy more the conditions that are representative of the most common environment, and that's going to drive how abundant you be, right? So to summarize, soil moisture is a key driver. The dry sites host more diversity, and it seems like most species are very well adapted to these systems. And while there is a very high turnover with soil moisture, we don't see that being related to traits as much, and that's probably because intraspecific variation is really high, and really what is mattering is the phenotypic differences within species that is allowing them to occupy some of these places and determines macroecological properties, right? And so some of this work tells us fundamentally how are species, or what is it that makes species common or rare? Can we find these properties that we can measure very simply and predict, is this going to be a common or a rare species? Because commonness and rarity is very closely related to extinction probabilities and how species might respond to changes in different situations, right? Which again, as I come back, is important for us to predict that if this climate or the particular microenvironment in a region is changing in a certain predictable way, can we use these properties of species to then predict how they will respond to these changes that are ongoing, right? A lot of this is very new work, as you saw, and most of this work was led by different master students who came into the lab. So I'm very grateful to them for all the hard work of working in these very, very tough conditions, doing these plots and generating all of this data. Hopefully some of these publications will come forth very soon. And I have to thank, this was from a grant by SERB, the Early Career Grant, and also funded by CCMB when I was there. So I have to thank those institutions and all of the students who did the fantastic work in bringing all of this together. And thank you so much for your attention.
We have time for a couple of questions.
Hi, Meghna. Really nice work. I had a question about whether any of your sites had disturbance gradients and if this affected the community diversity that you found.
Yeah, depends on what you call disturbance, like whether fire is a disturbance or not. In the sense, it is an ecological disturbance, but it has an ecological role. But the reason we chose this landscape is because it has relatively low human footprint. Right? So there wasn't lopping, felling, biomass extraction, or things like that.
No grazing also?
No, very little. No, no grazing.
Okay,
I won't
so
say no, but very little grazing.
and what about invasives? Were they in your plots or did that play any
Great
role in
question.
your-
We avoided plots with invasives, but then we started a parallel study in which we looked at, and we are still continuing it. There's a long-term effort where we are looking at lantana that has invaded some of these systems, replace the native understory grass, and we are trying to see how this invasive is changing the dynamics of tree recruitment and mortality, and mostly at the younger life stages. And interestingly, there we find that traits seem to predict the response of these simple traits like leaf traits seem to predict how species are responding to an invasive lantana.
Thank you for a nice talk. I'm a chemist and I was wondering about just one element figuring in your slides, that is phosphorus. And how about nitrogen and other things? Does phosphorus have any role in what you are doing?
Yes, so in many of these, so of course nitrogen and phosphorus are both key, are known to be key nutrients that limit plants in these systems, and by and large they have a macroecological value in terms of how communities are changing. So that's why nitrogen was also there, by the way, and we did have other nutrients as well. We also had pH and carbon nitrogen, but none of them were playing that important a role. It was primarily phosphorus and nitrogen that discriminated plots, and phosphorus from smaller scale individual studies of plants we know imposes the strongest limitation often.
Okay, thank you very much, Meghna, and thank you to all the speakers of this symposium. I think we had a wonderful time, and thank you very much.